Hello, everyone, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Today, our expert speaker, Brady Wu from Biointron, will be discussing developability assessment. Why, what, and how. This webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Additionally, please note that a few poll questions will be released at the beginning of the webinar and before the Q&A. So I will release those poll questions now, and I will also turn the presentation over to our speaker, Brady. Thank you.
Yeah, thank you very much, Christine. Sorry, I didn't show my face because I'm at a hotel and the setting is a little bit off. But if you want, you can see my face on the last page of our presentation. Yeah, today I want to introduce a concept of developability assessment and why we are doing that and what are the developabilities. That's a very wide concept and how we are going to perform the experiment. First, I want to show a story because I think storytelling is quite important. This is about a class of antibody. The target is PCSK9, so it's for metabolic disease. And there are already two. So I asked a DeepSeek question. Tell me about Pfizer's PCSK9 antibody products and why it was not approved. Of course, they have two. On this page, they have two antibodies already approved, one by Amgen, the other by Regeneron. But Pfizer's antibody, unfortunately, was withdrawn during a phase 3 trial, as shown on this page. And Pfizer disclosed reason as high immunogenicity, variable efficacy, and higher adverse events. Of course, this is every clinical project is quite complicated. There is no single reason pointing to the why to the causes, right? But since our topic is developability, I want to show you a little bit data later on how this antibody, which is called bococizumab, fared in the developability assessment. So why we are talking about developability? I think if you're working on antibody discovery and development, you can agree with me that we are entering the phase of the era of multi-specific biotherapeutics. Besides bispecific, specifically, we are talking about antibody cytokine fusion protein, ADCs, and right now even antibody oligoconjugates. So we are talking about a broader class of biologics compared to 20 years ago when we are just working on monoclonal antibodies. So we are entering the era that the methods to evaluate therapeutics and the criteria are becoming more and more difficult. So what is developability? This is a paper published on 2024. So when we are doing antibody discovery, we are starting with a lot of different molecules. So the number of molecules are huge. And we are doing a lot of assays to narrow down the number of molecules. But when we go forward on our program, the risk of our molecules are become bigger and bigger. So in this page, you'll see the green box called developability assays. They are a combination of different things, including I think the resolution is a little bit low, but you'll get the idea. The chromatography, thermal stress, mass spec, colloidal stability and manufacturability, and in silico testing. So when we are working on a large molecule, they have a native state, but they can go to other states, such as unfolded states, misfolded states, and aggregated states. So developability is a group of methods to evaluate the stability of the molecule, especially in formulation setting and also in vivo. So we have a very classic concept of developability. That's before or after formulation or pre-CMC stage. We are talking about the manufacturability, and we are talking about cell line stability, expression titer, scalability, stability in progress, in process, long-term storage stability, and comparability when we are talking about the biosimilars, and the cost of goods. So when you think about the antibody discard process, that's pretty later on. That's when you have a very few molecule, which you will choose one of them going to the CMC. Of course, we can use in silico methods such as hotspot benchmarking and chromatography, mass spec, and CE, and force degradation studies, but they are still before the CMC, and you probably have quite a few molecules. Sorry, just a few molecules that you will go to pre-tail studies in monkey. So that's a very classical concept of developability. So this is a paper published by a scientist. So specifically, we are talking about the CMC stage stability, including oxidation treatment, low and high pH, freeze thaw experiment, less stress, small stress in different temperatures. The reason is that right now we are working on a lot of biotherapeutics administered in very high concentration, close to 100 milligram per milliliter, especially for autoimmune disease and metabolic disease. For cancer, we have a slightly lower requirement on the concentration of the formulation. And so in that case, we have to do these experiments. So those experiments will tell you how the antibody fares when we stress them, when we apply stress on the antibodies. So that means you have a simulation of the antibody. So you do a 40-degree. That'll be a simulation of the conditions in vivo, or you do freeze thaw. That will be a simulation of the transportation and the storage. So that is a experiment looking for change, right? And if you look at right, so that's so this page is mostly talking about experiment looking for change, and that's very classical when we are talking about the monoclonal antibodies, and we have to perform all of them in order to have a very good pre-CMC package. And we have seen, as I said earlier, we have seen an evolution of biotherapeutics. That was just a very new review published by Genetic, the scientists of Genetic, and they are talking about the evolution of antibodies in the same indications or even on the same targets. So this is on the left, we are talking about the antibodies applied to blood cancer, CD20, CD19 targeted. We have firstly monoclonal antibodies and later T-cell engager with FC, without FC, and CAR T cells and nucleoside antibody conjugates, and also antibody drug conjugates. And on the right, we are talking about the HER2 targeted antibody for breast cancer and other solid tumors. And we are talking about monoclonal antibodies with engineered FC, and also paratopic bispecific with HER2 with targeting different domains of HER2 protein, and the HER2, HER3 targeted bispecific, and also ADCs. So if you look at the format of bispecific, the developability difficulties are increasing because we have more and more combinations and let alone CMCs. The process to make such molecule in preclinical setting is become more and more difficult. So the scientists introduced the concept of a discovery stage assessment because before we were talking about development stage assessment, including stability, chemical and physical stability, solubility, viscosity, and formulation. And since we're working on more and more difficult molecules, we want to move the development stage earlier, including in silico sequence analysis, thermal stability, and serum stability and specificities. So we all know that the antibody discovery is working like a funnel, especially on the functional side. Firstly, we are working on binding efficacy, and then we move to cell-based and in vivo and products and PCC. This is one side of the story. And in the other side, we are talking about solubility, stability, aggregation, and all of the different developability matrix. It's quite complicated, but we have in Biointron we have optimized the system to give you some very common options of the developability assays to give you a full evaluation of the developability. So in theory, this kind of in vitro assay will give you guidance on how your molecule will perform. For example, the thermal stability will tell you whether your molecule is stable enough in vivo, because if you have a molecule with a lower TM, that means half the molecule is melting. You can imagine the molecule in vivo will have a hard time, right? So we are introducing the developability assessment in early stage because before, when we are working on monoclonal antibodies, since they are natural antibodies, and mostly they are coming from immunization, either by mouse or humanized mouse or other animals, they are already selection pressure in place in vivo. So before, the scientists are mostly focused on functional selection. That means picking good candidates. But since right now we're working on more and more difficult molecules, we want to eliminate the bad candidates, that where developability is coming. So in this very conceptual figure, I draw a square that always called area of success. That means we are picking good functional candidates, but we are also eliminating bad developability candidates. So there are two dimensions selection. Okay. And I want to show you there are a lot of studies statistically showing a lot of different assays have a correlation with the PK performance of molecules in vivo. For example, the self-association index, which was performed by the assay called ACSINS, has a pretty good correlation coefficient, 0.55. And the Fc iron binding, which is a receptor for the recycling of the antibody, has a similar coefficient, 0.56. And the other one is non-specific binding, including PSR and BAP binding, has a coefficient of 0.67 and 0.69. That proves that this simulated in vitro assay focusing on the performance of the molecule, some physical chemical performance, and also non-specific binding performance will give you a very good guidance on how your molecule will perform in vivo or in human. And of course, you can start from in silico developability. This is a publication by the scientists of AstraZeneca in 2019, and they found that they grouped the therapeutic antibodies in two different groups. One is called a common group. The other is called clinical stage antibodies, and they found that in the group of clinical stage antibodies, clinical stage therapeutics, which is called CST, they are more converged in terms of several characteristics, including the surface of the hydrophobic surface, positive charge surface, and a negative charge surface, even the CDR length. So they came up an index called a statistical index for therapeutic index for the antibodies. That was purely sequence-based in silico developability prediction. So that will give the idea when you have a lot of sequence to work on without any YLab, you will find some of the better candidates already. And this is a publication by Adimap Science, and they found that if you look at several different assays, PSI means polyspecificity reactive assay. The approved antibodies has a significantly lower PSI score. That means the approved antibodies compared to the phase 2+3 antibodies, they have much lower non-specific binding and much lower PVP binding, which is also an indication of non-specific binding, and much lower analytical aggregation. So that was what AS means. So when we are going through the clinical stage, this is there was a natural process that the bad antibody will be eliminated. Right now, we just want the process to be pushed forward as much as possible, because the in vitro assay has very low cost. But if you found a good bad antibodies in rat or in monkey or even in human, that would be very expensive. So they also did a study to build a correlation of different developability assessment, because they have a lot of different methods. But as I said, we don't have a lecture to run all of them, but we can do some of them. And there are some applications of developability, especially in the field of AI. In this page, I'm showing that we have this progress on the left of page, discovery, developability, and formulation. And we can have a machine learning algorithm embedded in the drug discovery process. When we have good data, when we have good automation, and the data quality is high, and we can have the data inputted to build some correlation between the sequence and also their fundamental characteristics. So that's one way to combine the AI and developability. There is a other way, because right now we have some programs to generate the antibody sequence de novo. And in that case, we need data and a model to train our algorithm to generate a better sequence. So one way, on the last page, we are introducing the AI as an assistant for the drug discovery process. But in this way, we are generating data to assist better AI to, again, to design the sequence de novo better. Right? So I just show you one example. It's a really new publication. They did a data set of about more than 100 antibodies, and they used that training set to build the correlation of the sequence and the correlation of sequence and the melting temperature. And they have a pretty good model from deep learning to do that, and they can predict the melting temperature of the antibodies beforehand, before any wet experiments. Of course, the correlation is not perfect, but it's good enough to narrow down the candidates. That's one application of the developability. And this is another. The slide is quite busy. It was a really new paper published by a Merck scientist. So they have done developability for bispecifics, protein A titer, SEC purification and SEC purity and CSDS purity to group different antibodies, because different antibodies will have different characteristics, and they can pick good antibody with good functionality within the same group. So that's for bispecific, and they apply the similar principles on the monoclonal antibodies, and they did melting temperature, aggregation temperature, Higgs rotation time, and PSI ELISA to group monoclonal antibodies. So in that way, they will find okay, this kind of antibodies are quite similar from the immunization library, right? So that will help them to select better candidates. And this one, again, it was a very new paper because, as I said before, right now we are working on antibodies with very high formulation concentration, more than 100 milligram per liter. And again, this antibody before, that was not very good antibody because the viscosity is quite high. And they try to find the self-aggregation site where hydrogen deuterium exchange mass spec, and they found the site and did some structure prediction and did some mutations. And the important thing is that they run a few assays. One is assay sense to get a score of self-aggregation probability, because this assay will give you a score of self-aggregation in very low concentration. And they did other developability assay, including thermostability, non-specific binding, and also DLS for aggregation. And they found a very good correlation of their mutants between the viscosity score and also the aggregation profile in DLS. And on the right side, the figure C showed the very good correlation between viscosity and the ACC score. So so this is a method to narrow down the candidates before you really run the viscosity experiment, because in that experiment you need really high concentration of antibodies. So there are a lot of developability assays which can help you narrow down the candidates to do because before you do the monkey experiment, probably you will generate multi-hundreds or even grams, multi-hundreds of milligram or even gram of antibodies. So this is a method to give you a sense with very little amount of antibodies. As I just showed you a couple of slides before, you just need a couple of milligram of antibodies. So this one showing your application is quite interesting, because in this paper, I think it's published by Oxford, they use a AI language model embedded by the work by AstraZeneca. So you may remember the score of hydrophobic surface, and a positive charge surface and a negative charge surface. And they want to prove that they have optimized the model to give you very good in silico developability score. And then in that way, you can narrow down the sequences before doing any Y-lab. Yeah. So it's really interesting work. So this is another work. It's generated by de novo. All the antibodies are generated de novo, and of course, you want to do some binding experiment by fate very quickly, and then you want to do all kinds of developability and functional assessments. And I just showed one set of data. That's AC-sense data, and again, you see a green box. That means all the candidates here is safe, and all the candidates above the green box will give you a flag. That means it's not a very good candidates in terms of self-aggregation probability. Yeah. So this is just another application of the developability in literature, and this is a paper published by Chinese scientists last year. They did a bispecific common light chain antibodies against PD-1 and PD-L1. They did a bunch of developability assessment, as is HPLC, multi-temperature hydrophobicity, and CESDS and CIEF, and so this kind of data, of course, they have a lot of candidates, but they just show several candidates for the development data. But this kind of data will help you select, will help them select the good candidates besides functional evaluation. So let me go back to the question. So what went wrong about these antibodies? So we want to test how the antibody is performing on the developer assessment experiment we set up in-house. Of course, we often got asked that some antibodies which got very bad developability was approved or in late clinical phase. So how can you justify the necessity doing developability? My answer was that it's like you are picking a basketball player, right? You want a very tall guy to play basketball, but sometimes a short guy, such as Allen Iverson, would be a very good basketball player. So there was a chance, but under the same condition, if all of them are playing, have the same technique and same skills, you probably want to pick a higher player, right? So if you have a group of antibodies which got very similar functionality, but some of the candidates had very bad developability scores, probably want to eliminate them first and pick others. So that was really the meaning doing developability so early. So let me get back to this question. What happened? So we are establishing the PSR-ELISA method in-house, and the bococizumab was actually a positive score in our setting. It has very strong PSR score in 3 different regions: baccalaureate particle, double-strand DNA, and insulin. So it has quite strong polyspecificity. That's one. And this one was AC-sense experiment. That's showing you if you have a very large redshift, that means you have a very strong tendency to self-aggregate. So again, bococizumab was quite high in this list. Actually, it was second or tied first in our setting. Again, very strong tendency to self-aggregate. But when we look at thermal stability, it has okay thermal stability, because if the score was about 65 degrees centigrade, we will treat that as a good candidate in terms of thermal stability. And the hydrophobicity, we normally use trastuzumab as a control of hydrophobicity, and bococizumab has a slightly worse hydrophobicity score because it has it was more hydrophobic than trastuzumab, but still within our range. We will think if you have a score, relative retention time larger than 1.5, that means you have a bad antibody. So let me go back to the conclusion I made personally. So this antibody, bococizumab, it got good thermostability, okay hydrophobicity, but very strong self-interaction. That means it has very strong tendencies for aggregation in vivo. And it has very high PSI ELISA score. It was a positive control for our assay. That means it has very strong tendencies to have off-target binding and higher immunogenicity possibilities. But we don't know because it was just an in vivo assay, right? So I just want to show you how we apply the method in-house, and we can do a hydrophobicity ranking, especially for ADC, for antibodies for ADC application. Since the small molecule is usually hydrophobic, you want to measure hydrophobicity. If they have the same characteristics, functional characteristics, and other similar developabilities, you probably want to pick antibodies with good hydrophobicity. And this is just a ranking of PSR ELISA, and we can show you some of the antibodies has very strong BFP score and was red flagged. So that will be eliminated first, and the others will be kept. And this is just two antibodies to showing whether they have any chance of self-aggregation in low concentration. That's AC synthesis. And we have a positive control, which called infliximab, and has a very large redshift. In this assay, if you have a large redshift, that means this antibody has a strong tendency to self-aggregate. So fortunately, these two antibodies were quite good in this assay. Just to let you know, infliximab, of course, it was a very famous antibody, and it was approved, and it was a blockbuster. But when we built the assay, we saw the data, and we saw the data in the literature as well. And so we wish to find an explanation on why this antibody has a very high ACC score, and we checked the literature, which said this antibody need formulation optimization. So that gave us indirect evidence that this antibody might have some strong self-interaction and need to be optimized in formulation. So I want to give you a summary because I was telling a story today. I didn't focus on each assay, so I will have about a dozen of different assays. But I just want to tell you that what is developability? Developability is a very classical concept. But firstly, it was introduced along with formulation, stability, and SMC. And since we are working on so during the antibody discovery development, the concept is widened and it contains more comprehensive information, including biophysical characteristics and also non-specific interaction. And so we use instrument and also ELISA plate-based assay to do developability assessment. And since we are working on biospecifics, ADCs, and more complicated molecules, I think it's quite important to push developability forward to lower the risks as low as possible. Before, we were working on monoclonal antibodies and was naturally selected, but bispecific antibodies were mostly human-made. Right? They are products of engineering. So the developability assessment will be more important for bispecific compared to monoclonal antibodies. So along with functionality, developability forms a multi-dimensional assessment system for your PCC selection. Of course, AI is a big thing, and we have seen the trend with the improvement of AI. The data generated with the developability assessment can be used to train your models, and even further accelerate the drug discovery process. So this page shows what we are offering right now. We have the QC for developability samples, because there was a thing called garbage in, garbage out. So you want your samples have good qualities. And we have a package called early developability, including thermostability, TM and T-onset, PSR, ELISA for non-specificity interaction, AC for self-aggregation, HIC for hydrophobicity, TAG for aggregation in formulation buffers, and also rhythm analysis for any chance you will get soluble aggregates in solution. Also have some other assays, including heparin HPLC, which will tell you whether your antibody will be positively charged, and also some late developability pre-formulation, including ion exchange, HPLC, and CIEF. Of course, we are expanding our package to serum stability and PTM analysis, and also pre-formulation stability testing, or if you are looking for data for change. And lastly, I'll take a few minutes to introduce who is Biointron. We are a CRO company focusing on preclinical antibody discovery and production. We have more than 600 employees, and we run about 100 discovery projects per year, and we generate about 100,000 antibodies per year recombinantly. And so we have antibody discovery service for APACA and single B cell technology, expression, optimization, including affinity modulation, harmonization, and bispecific antibody production. We have produced more than 260 different formats, and we have antibody drug conjugate service, and also developability evaluation, including, I just mentioned, thermostability, hydrophobicity, non-specific binding, and also gram-level antibody production. We got a PACA form, self-owned in China, more than 300 alpacas, and we breed them in very early age, and we breed them more than 1 year before we took them for immunization. Before we take them to immunization, and we have a quite automatic lab, and we have clean room, molecular room, ACTA shaker, and a bioreactor. That's very common practice in mammalian expression labs. But we have our edge on automation, just showing you a line of plasmid prep automation. We have a cell dispensing for E. coli, plasmid prep, and also a very big machine for pipetting and labeling. But we have automatic investment on other parts as well, including transfection, purification, and quality control, and analytical method development. So that was about it. And if you didn't see my face, this page will show it. And I'm happy to take any questions.
Thank you so much, Dr. Wu. So we had a couple of additional poll questions at the end, so I'm going to send those out really quickly, let people respond. And while we're doing that, if anybody has any questions, you can add them to the Q&A. I see we already have a couple. So the first one I can answer because it's about slides. This question says, is it possible for the slides to be shared after the seminar? And as long as Dr. Wu's okay with it, we can upload the PDF copy of the slides to the viewer, so everyone will be able to access it. So while we're waiting, because it looks like some people are filling out the poll, please add more questions to the Q&A. The first question for Dr. Wu is, how do you detect hydrophobicity?
Yeah, we use the HPLC method. So we have a column with a short or long carbon chain, and we run the experiment in the HPLC machine. So it's like SEC but different columns. Yeah.
All right, thank you. And now we have a few more. In your slide on AI and developability, can you comment on the FC effector function piece?
Yeah, that's a very good question. I didn't mention that because sometimes people will consider FC function as a part of their developability, especially for FC iron binding, because it's directly correlated to the half-life of your antibody in vivo or in human. And but there are also other mutations affecting the binding of FC receptors in immune cells with antibodies, right? I think you're asking that. I have seen a paper showing that those antibodies, especially for the mutations, which will decrease your binding to FC receptors, the antibodies will give you a slightly lower thermostability, but not very big change. So that means the durability is still okay. Yeah.
Great. Thank you so much. How do you detect off-target binding?
There are quite a few methods to develop off-target binding, such as true cell lysis, or I have seen ELISA-based method and also FACS-based method. But what we developed was a baccalaureate particle-based method because we have a baccalaureate group in Biointron, and we can get the virus very easily, and we use several different reagents. A baccalaureate particle was a good simulator for the conditions in vivo because it has membranes, it has DNAs, and it has a lot of different proteins. So if you have a very high score on this assay, that means your antibody should have very strong non-specific binding. The other assay we introduced was double-strand DNA binding because DNA was quite negatively charged. So that was a good simulator for the conditions in vivo when your antibody encounter membranes or circulated DNAs. Yeah.
Okay. To what extent do you use the data you generate to develop your own AI models?
Let me see. Actually, we don't have our own AI models for drug discovery purpose. I think when I mentioned the AI models in the slides, I was talking about the method you want to build the correlation between your sequence and the functionality, such as binding of efficacy, cell-based binding, and non-specific binding. But we don't have such model. We have models for codon optimization, but that was for protein production, not really for functionality because we are a CRO company, right?
Thank you. Next, how do you assess developability of bispecifics? By the parental maps?
Yeah, that's a very good question. So I think you want to evaluate the developability of your parental maps, but since bispecific antibody is one molecule, and That's quite obvious, right? So you want to combine the two antigen binding arms on the same molecule and to evaluate the developability again. As long as your antibody has a FC, most of the developability methods developed for monoclonal antibodies will be applicable for bispecifics as well.
Got it. Thank you so much. Next, which protein would we use for the off-target protein? Is it the same family-based protein?
That's a very good question. I think we are talking about two different things. One is polyspecificity. That was you are talking about, because that means, for example, if you are developing a antibody against choline 18.2, you have another protein which shared very high homology, choline 18.1, with your target. So of course you want to do some off-target screening against antigens with very high homology. So that's called polyspecificity binding. That's called polyreactivity binding. Sorry. Yeah, that's quite complicated. So you want to test the antibodies against other antigens with high homology, right? I think some of vendors have very comprehensive assays for all the membrane proteins in human cells for that kind of assay. But what we are talking about is a non-specific binding or polyspecificity. That means the binding of the antibody against any dirty targets. That was not very strong, but that will affect the half-life of your antibody in vivo, because in vivo you have very dirty experiments. You have environments. You have membrane, you have DNA, you have serum albumin, you have other IgGs, you have insulin, you have a lot of different proteins. You don't know what your antibody will bind to. So we have this in vitro assay introduced to give you a sense whether your antibody will have a high chance to bind to any dirty antigens in vivo. Okay. I think you are muted, Christine.
You're so right. Sorry, I muted myself. I'm sitting outside, heard an ambulance driving by. So we have one more question. For antibodies that fail clinical trials due to developability, is it primarily a result of the CDR sequence or other regions of the construct? For example, FV framework, FAB constant regions, FC, etc.
Yeah, that is a very good question. Actually, I would say that's a combination of everything, because again, we are talking about one molecule, right? There are all the different amino acids are covalently bound via the amide bond, right? So it's not like only the CDRs or framework or the FCs will determine the fate of the antibody. I think it's everything. So it's quite difficult to answer, but you can use a lot of different programs online for in silico prediction, and also you can use developability assessment to evaluate your antibody comprehensively. That, I think, is really a multi-dimensional question. Yeah.
Thank you so much. And thank you, audience, for your great participation. I'm gonna pause for a second to see if we get any final questions, and if not, I will wrap us up. Okay, I'm not seeing anything else come through, so I will say thank you so much again for this insightful presentation, Dr. Wu. The on-demand version of this webinar will be available on YouTube shortly, and a link will be sent by email to everyone who registered. And as I mentioned, we're also happy to share the PDF copy of the slides. And you're able to watch this webinar or any of our other on-demand webinars whenever it's convenient for you on our YouTube channel. Thank you again and have a great rest of your day.
Thank you, Christy.